you very much for the opportunity to be here. I'm very happy to be in Vienna. I did my postdoc in Prague, and walking around here is like a flashback. It looks very similar in architecture. The pastry is the same. I'm going to present this work with perhaps a slightly cryptic title, and I could maybe say that this is a little bit of accidental research because we actually wanted to do something else, but we ended up doing this. And I will try and explain a bit along the way what we actually did. And it's going to be a bit technical along the way, but I'm also trying to put in a lot of applications of why we're interested in doing these kind of things. So what I'm interested in is complex systems where we have macromolecules. It could be proteins, like we've shown here, that interact. And usually those proteins are not simple spheres, but they are irregularly shaped bodies, and the interactions between them is directional forces. And those forces are very important for the properties of the solution. So the first one here is a snapshot from a simulation of many lipase molecules. So this was together with a biotech company in Copenhagen, and they add lipases to dishwasher fabric detergents. So the lipases, they feed on grease, so they actually help degrade and clean the fabric. And they are interested in how they interact and how they interact with surfaces. Study where we compared Monte Carlo simulations with the SAC study, and this is exactly an application where you need to treat many molecules and the first day, Peter Schurdenberger, he showed colloidal systems. We have similar kind of modeling. So the length scale here is not important. It could also be way smaller, like on single molecule scales, but it's basically anisotropic interactions that we are after. And when we talk about protein-protein interactions, I think it's important sometimes to just explicitly state that it's not really just protein-protein interactions. We also have to consider the interactions with the solvent and co-solutes and all these questions. We really need to include all of these effects while describing the protein-protein interactions. And in an ideal world with perfect force fields and infinite computer power, we could just do atomic level simulations and walk around here in this kind of circle here. But unfortunately, if we are going to study things like I showed before with perhaps a couple hundred proteins, the length and time scales we have to cover is these models, and also the many parameters we have here are difficult to handle. So often we have to remember that when we go to more coarse-grained models, we still need to include all of these effects of the solvent and the co-solutes. And I think we've seen many examples here during the conference, how we average out certain degrees of freedom. Doesn't mean that if we go to implicit solvent, doesn't mean that there is no solvent, it's just implicit, right? And when we have implicit salt, it's a Poisson-Boltzmann. And we've seen examples from David, for example, trying to include some ion-specific effects into these models, because when you cross from an explicit solvent to an implicit solvent, many of these specifics are lost, and we have to somehow bring them back in. And I'll briefly show here 3 different effects of salt, how they might affect protein-protein interactions. We've seen binding ions. This goes into the charge regulation, Hofmeister effects. That's up in this category. Of course, we can screen the charge with the device screening length and so on, so we can the length of the electrostatics. But we also have these kind of effects where some ions, they're actually excluded from the macromolecules. And in the Hofmeister or ion-specific terminology, that would be sorting out effect. we also have to deal with that some ions, they're actually staying away from the molecules. And the fact that they do that, that can push them together, so we can have an attraction between molecules due to that effect. All of these three effects I've mentioned here can lead to anisotropic interactions in one way or the other. And that's kind of the realm that we're interested in, how the solution properties affect the orientation and thermodynamic properties. So this is an example, and this relates a little bit to Natasha's talk about macroions in multivalent salt. So, and I'm now also introducing a model, this charged patchy particle model by Joachim Dubiela, who was here in Vienna, I guess many years ago now. And it's similar to Sebastian's model, where it's kind of a toy model for a protein, but it's designed in a way that it has charge density and is similar to a globular protein. And the nice thing about it is that you can systematically change the patchiness, so you can have two patches, one patch. But it's kind of a way to extract and wrap your mind about complex anisotropic electrostatics in proteins. It's an artificial model, and we will get back to, let's say, more real proteins later on. So if you normally, these particles I've shown here, they have a net charge, but they also have a dipole moment. And if you put them in solution, they will normally orient like this. So the dipoles are pointing towards each other, or like that. So there's an attraction. But if you add trivalent ions to the solution, you can actually make them aggregate like this. You can have these kind of correlation effects, and this is something that you can introduce by adding ions. This is a bit, perhaps a little bit of an exotic example, but people have studied trivalent ions and proteins. There's a group of Frank Schreiber who has done a lot of that, and there are some interesting, peculiar features of that. Maybe you don't want to have these trivalent ions in your body, but for a model system, they're quite interesting. So, and now we're getting to we're getting closer to what I want to talk about and the motivation for what we have been doing. And that is that when we want to calculate and handle these proteins or macromolecules in a computer simulation, let's say that we have our two proteins up here, and we want to calculate the interaction energy. They're in a certain pose, or there's an alignment that has a set of angles that describe how they're oriented towards each other. And then we have some interaction sites here, and we are simply just summing that up, get an interaction energy. And now we are talking about some sort of and if typically for proteins we have maybe around 1,000 interaction sites, so this means to calculate a single pose, it's around 1 million operations. If we don't use tricks like cutoffs and so on, but when you go to low salt concentrations, you have to be a little bit careful with cutoffs because you have long-range interactions. Let's now say that we have many body simulations, and I've shown you a picture where I think we had maybe 200 up to 500 proteins. Then we need, for each one of those pairs, we need to calculate their interaction, and we might have in the order of hundreds of proteins. So I think you can quickly realize that this is going to be expensive, and but still needed if we want to, for example, connect to our experimental colleagues doing SAX experiments. We often want to go to these systems, or if you want to study phase transitions, it's heavy if you want to treat it in that kind of representation up here. So this is these are based on PDB structures. Take the crystal structures and put them into the simulation. Of course, we can start to cross-grain these further on, right? And we've seen many examples of this. You can also go the other way, right? You can start from a sphere and start to make that more patchy. Here we go the other way. We start from the atoms and then we less patchy. So the idea is that if we can calculate this. If we can pre-compute these, we can actually speed up the simulations enormously. And Harold Hatch here from NIST did that recently on antibodies, and it's particularly when you go into the antibody systems where you have maybe more than 1,000 atoms in each antibody, you can see that this could be spread out by orders of magnitude, recalculating these interaction between poses of the proteins. And we're getting closer now to what we want to do, and this is idea that we want to try and see if we can pre-calculate the energies in this ending up being a 6-dimensional space. So we have the distance between them, but then we also have 5 angles, and we can do that in many different ways. And if we want to build up a 6-dimensional table, lookup table that we can use in computer simulations. I'm saying that, but we actually ended up doing something different. But let's get back to that again. So if we just look at 2 angles here, let's just look at a simple protein, and let's say we want to tabulate properties on the surface of the protein. So what we could do is that we could make a matrix, and we could just distribute these 2 angles linearly on that matrix. And that's generally a terrible idea, and it's not surprising at all. But if you do that, your points are going to look like this on the unit sphere, so highly unevenly distributed. So generally, we don't want to do this. You don't see that, but up here on the poles, you actually have a lot of points overlapping each other, and it makes interpolation on this very difficult. Instead, what we can do is that we can use subdivided icosahedrons. So we start with an icosahedron, and we can then subdivide it further and further, and this creates a very nice uniform grid, and it has some nice properties for tabulating that we can use. We'll get back to that. If you have any interest in this kind of systems, this is not something I knew so much about while we started, but the book on this is very fascinating. You can use this for all kinds of things, for optimizing traffic to lots of different things. And it's a fairly new book and a very interesting read. And a lot of these algorithms for handling this comes from computer graphics. So it's used a lot in gaming. They do this kind of stuff all the time. And this also means that we can find nice libraries. So if we go back to our charged patchy particles, and I'm just going to show you how it looks if we try to tabulate the electric potential on the surface of this. And you can see the points we have here. They correspond to vertices on these subdivided icosahedrons, which nicely covers the unit sphere. And all the other points, the colors here, they're interpolations between those points. And you can see that in this linear, I mean, they are distributed quite irregularly on these angles. The unit sphere is completely regular. Interpolation between them can be done with something called barycentric interpolation, which is done on the faces of the subdivided. Also super convenient and a nice properties for handling this. But 2 dimensions is not enough. We want to go to all the 6 dimensions, and the idea is like this. We have our 2 bodies. We have 2 angles here. We have an angle here and 2 angles here. The way we do it is that we create a regular grid on this omega and the distance, and on this grid we place the subdivided icosahedrons, and on the vertices of those we place another subdivided icosahedron corresponding to those angles, and on the vertices of this is where we store our data. And in this case, if you want to build a table we can use in fast computer simulations, this would be the interaction energy. We can now subdivide this further if we want to increase the resolution, and of course it gets more and more expensive. We have more and more points we need to fill by doing that. I should should also say that this subdivision doesn't have to be regular along, for example, the distance. So if you are at long separations, you don't need to do any subdivisions, but only when you get to close separation. You could even imagine that depending on, I mean, you don't even have to have same number of subdivisions regularly on the surface of these. So there's room for some optimizations here. And yeah, now this is now we programmed this, and you can almost imagine that there could be many ways where you could make mistakes in programming this. So so we started to think about how can we test this? How can we verify that we did this correctly? And what turned out, and what we quickly realized, and this is not something we thought about from the beginning, is that having done this, having filled out this table elaborately by calculating all these interactions, is that we actually now can brute force calculate the partition function or the configurational integral. And this is something that we almost never do in statistical thermodynamics because it's just so expensive to do. But here we had it for free because we had already calculated it. And then, of course, having done that, you can do whatever you want with it. There is now it's of course discrete angles, and there is a degeneracy here, and that has to do with the size of the triangles surrounding each vertex. And if you have no subdivisions, that's fine because it's a constant. But when you start subdividing, there start to be slight fluctuations in these triangular surfaces. Calculate that, and it's not a problem to handle. Having done that, we can now calculate things like the osmotic second coefficient, which is something that we can go to our experimental colleagues and ask them, how does this fit? And this was a complete side effect that we didn't think about, and they actually deviated into that kind of, we went more to this B2 calculation, and I will show you some results and also some reflections on what we found doing that. And I would like to show just how it works. And this is a small video I did on my laptop here, so yesterday night. And so the program here is it's available if you want. And you just give it two structures of your coordinates. You need to say what kind of interactions you want, the angular resolution. We have an ionic strength and interval you want to sample, and so on. And run it, and you can see it's a very now we have calculated around 500,000 poses, and you get kind of here we get the potential mean force, and we can also calculate the average energy, yield coefficients, binding constants, and so on. Don't mind these silly molecular mass, it's just the way I set the weights, but it doesn't really matter. So very quickly we can calculate these properties, and comparing to simulations, it's very robust in that we don't have to worry about the Boltzmann inversion of a G of R or anything like that. You get the result immediately, and you can actually automate it. It also parallelizes trivially, so you can run it on as many cores as you want because each operation is independent of each other. We don't have to worry about deep free energy minima like we do in simulations because we just brute force calculate it. We can, of course, compare with Metropolis Monte Carlo simulations, which generally tend to run slower because they're difficult to parallelize for these small systems. But we get good correspondence. So this tool is sort of the where we ended up, and I'm now spending the rest of the time just telling a little bit of the result we got from. So because it's so cheap to calculate, we can now take these particle here and calculate for a lot of different points osmotic second virial coefficient. And what we have here is the temperature, and what we have here is the ionic strength. And if you go along the ionic strength, you can quickly see that there's a non-monotonic behavior. So we have repulsion at low salt, go through an attraction, and then repulsion again. And this is something that we have observed before for proteins with light scattering and simulations. What I was really surprised about was that when you go in this direction, it actually, when you heat up, this gets stronger. And the reason why we see an attraction here is because of you have a dipolar attraction that overcomes the monopole repulsion in this kind of system. And the reason I was surprised was that when you increase the temperature, I thought the Boltzmann, the angular distribution would be washed out, so that all the angles would have this patchy attraction would be washed out when you increase the temperature. And the explanation is quite simple, and it's simply because the arm length is an increasing temperature, increasing function of the temperature because of the dielectric response. Temperature. So if we artificially kept the dielectric constant, this would flip around. So one should never forget to take into account that the dielectric constant is. So there are some, and now of course we can explain what's happening in here. I'd like to now talk about real proteins and again osmotic second virial coefficients. And here we have a protein called chymotrypsinogen, which is I think it's maybe around 100 or 200 amino acids. It's not that big globular protein. And you see that kind of behavior when you have the ionic strength. It goes through a minima here at very low ionic strength, and it's because it has directional interactions that causes that you have directional electrostatic interactions in addition to a monopole. So what we try to do now is that we are taking a fairly recent force field use in the IDP community called Calvados. I don't know if you've heard about it, but it's from our colleagues in Copenhagen, and they use it for intrinsically disordered proteins, and it has been trained on radius of duration. We just misused it and say, okay, how does this work with globular proteins? And the answer is not very well. Well, I shouldn't really. So out here at high ionic strengths, and this is actually where I think most of their training data comes from. It actually is not bad, but in here we completely miss this. Here. And the reason for this, and I think that Peter talked about this at the previous meeting, and the model is simply one bead per amino acid, and it can be charged or neutral, and they put the charge here in the center. This means that potentially when we get a low ionic strength and into close contact configurations, we might underestimate it. So what we can do instead is to just off-center it, keeping it where we found it in the crystal structure. To do that, we can have a model like this. But we could also, if we don't want to move our charge, we could also say it sits in an implicit spring, just described by a polarizability here. So this is some sort of R4 term. So it's an ion-induced dipole term that we can just add to the potential in addition to the Coulombic interaction between the beads. We see that we start to now see that we can get this kind of quantitative behavior. So this indicates that there is some, so this definitely is that we have alignment. And we can see that because we have access to the full angular space, right? We have all these angles, we've stored them in our elaborately developed table. So this suggests that, that keeping them in the middle is perhaps not the best idea, and we can actually do a small correction by just off-centering it. And if you think about a protein like lysozyme, which is purely repulsive, this has a very small effect because the close contact configurations are not really populated. There it has a very small effect. If you should say something, I don't know if this is generally true, but it actually gets a bit, this model gets a bit closer to the experiments. But on the other hand, they are, it's not, osmotic second virial coefficients also experimentally, our experimental colleagues, they also admit that it's difficult to measure this and reproduce it. So it's not set in stone, I would say. We can do one more thing and then I'm done. So this is of course a completely solution properties, right? Infinite dilution, but we have a bit extra information because the experimental crystal structures of chymotrypsinogen, where we took the structures from initially, we can actually go in and look at the contacts. And since we have access to all the poses, all these our 6D table, we are now looking in the free energy minimum and just look at what kind of configurations do we have, and we are using a principal component analysis to cluster them according to similarity and if they have an attraction. So they're weighted by their Boltzmann weight. This is what I'll show you here. So here we have the principal components. We see that we cluster. So each of these peaks here, they correspond to attractive configurations that are similar but and have a very low energy. And the stars that we see, they are they mark the crystal contacts. So there's nothing in the model that tells us that we should recover those crystal contacts. It only comes from the force field. There's nothing that we have put in there, and it's I was actually I didn't know at all if this was going to work, and it seems to work. You recover those. Of course, we also see many other configurations, and that's maybe not surprising because these are from a crystal and this is from a solution. Right? And we can do the same for the alternative models that we have created, and we see basically the same distribution of this angular space, but the relative, you can see that we strengthen these. Scale here, it's when we do this analysis, we get a histogram of how many configurations we hit in each of these clusters, and now just taking the log, so you can see this is kind of an AT scale that we have here. But so we just simply by doing this, we change the process, you could say. So that's about it. A bit of a summary. So the real reason we wanted to do this was that we wanted to speed up simulations, but we just haven't gotten to it yet because we got stuck in this B2 business and the fact that we could calculate the configurational integral by brute force. But the method here has a lot of nice features for actually speeding up simulations and tabulating. For example, interpolation is simpler because we can use this barycentric interpolation. There's also the possibility that we might populate it with Metropolis Monte Carlo, so you don't fully populate the table, but just partially. All the high energy configurations, maybe we can skip them. We haven't looked into that so much. Software here, if you're interested in trying it out, it works for all kinds of, it doesn't matter, just rigid bodies, you could say. And we have this correction that we think might be useful for the Calvados model, and we are in talk with the group in Copenhagen to see if they could redo their training set. Yeah, and we can recover the crystal contacts and also connect to osmotic second coefficients obtained from. Thank you for your attention.